Thank you. Yeah, so this will be hopefully an introduction to pangenomics and particularly why I think they're worth exploring. As a kind of pre-warning to some of this is the type of pangenomics that I do. So I'll probably talk a lot about cattle because that's the system I work on. But many other species and systems will broadly be similar. Of course, there are many species that have their own unique traits where many of these topics may not apply or apply as easily, but hopefully this will work for many people. Genomics itself, so the linear reference genomics, has been and will continue to be successful. I think pangenomics are great and again are worth exploring. But that's not to say that not using pangenomics is an inferior technique. We'll also see that pangenomics is a very popular and growing field, but is, in my opinion, wildly undefined. So I might give definitions that are not the same definitions that you've seen elsewhere. And again, that's just part of this field being rather new. It's also fair to say that a lot of these topics change quite quickly, and there are many things that I feel like I understand, and then I'm completely disabused of that notion. So if you find this talk to be slightly confusing, just know that you're not alone. I also get confused by many of these topics, and hopefully a bit more exposure, and we'll get a better understanding of them. For the learning outcomes, hopefully by the end of this talk, we'll be able to, again, give some definition for what a pangenome might be, and particularly how we can distinguish it from a linear reference genome. And we can understand some of the different biological or computational motivations for why we want to work with pangenomes. Again, I think they are worth exploring, so hopefully we'll give some cases of some nice opportunities for what we can do with pangenomes, as well as some of the challenges that we encounter as we actually apply them in research. And as a quick overview, I'll again hopefully motivate the need for why we need pangenomes, talking a bit about basically genome assembly in the modern era, as well as different limitations of a linear reference genome, give a little bit of details on pangenomes, and hopefully some examples of where we can use them, and then finish on some exciting opportunities and some unfortunate challenges that we encounter with them. So some basic definitions that we can take from genome.gov. A genome is just the entire set of DNA instructions found in a cell. When we talk about a genome, we typically talk about a single linear reference sequence. So even for something like human, which is a diploid genome, we essentially will talk about a human genome as a single linear reference. Of course, the reference genome here is defined as an accepted representation that is used by researchers as a standard for comparison to DNA sequences generated in their studies. Key thing here is that, of course, every individual will have a different genome. Again, even for a human with a diploid genome, your two genomes will be different. We talk about a reference genome, so we can all talk about the same sequence. We can all talk about the same coordinates, just an easy way that we can make sure we're doing comparative studies in the same way. Of course, as we talk about genome assembly, this has gotten much easier in the era of long-read sequencing, particularly in the era of accurate long-read sequencing. Certainly for cattle, this is a done problem. I believe many of our colleagues in plants do not have it quite as easy yet. And of course, assembly is getting better and better every day. So even from this paper from a few years ago now, it really talks about how much faster and cheaper and easier it is. If you get long-read sequencing, you can basically produce a top-of-the-line assembly more or less within 24 hours even on a modest computer. This really has become a decent solution to sequencing everything. And of course, in the past, we really were trying to focus on getting a single genome to represent a species or even several species. Nowadays, we can talk about generating dozens or even more assemblies of a very similar population, species, whatever. Sequencing has become so easy that we can really do this for as many individuals as we want. And if we look at publicly available assemblies, so this is taken from mammalian genomes on NCBI, that's greater than 1 gigabase. We can see here that on this log scale, basically all the assembly types, so different levels of contiguity, are increasing rapidly. And this is, of course, also just public genomes, so there's many more unpublished genomes. So this is likely a large estimate. But we can see the number of genomes across, again, same species or across multiple species is rapidly growing. So we have far more assemblies accessible today than we have had in the past. And again, even some of the terminology I've used so far is a bit shaky. So when we talk about species, these reference genomes typically represent maybe a genus, maybe species. Of course, many species that we work with might have very distinct subspecies, might be very distinct populations. And for those who work in something like bacteria, there might be quite different strains. Even if you talk about clones, they might have slightly different genome sequences due to de novo mutations or so on. And if we ever talk about trying to have a specific reference genome, a single linear reference genome to represent an entire subspecies, population, strain, whatever, it's kind of this XY problem where the symptom that we're describing is not actually the underlying problem. We can never have a single linear reference genome that will suitably represent the variation in any non-clonal population. And this is actually a very similar problem to metagenomics. But we can talk about how there's many different genomes in this pool. In this case, it might again be some different subspecies or different populations, but there's no single way to represent the diversity using this single linear reference genome. So again, at least in cattle, we typically talk about something like the nearest reference genome to our sample. So the ancient ancestor cattle was auroch, then splits into the taurine and Indocene subpopulations. So if you have some sort of taurine cow, great, you can use the taurine reference. If you have some sort of Indocene reference or Indocene sample, great, you can use the Indocene reference. But biology is rarely so neat. There's many cases where there might be something like admixture, hybridization, introgression, whatever, where it has some sort of complex biological background. And there's no single way that we can say, okay, this admixed sample, we can use the taurine reference because it contains sequence from a different breed or a different subspecies. And again, there's no single linear reference genome that we ever find that would represent this other than the animal itself. This becomes a bigger problem when we talk about something like reference alignment, where when you're doing sequence alignment, it's very easy to confuse mismapping, so a breed mapping to the wrong location, with just genuine biological divergence that your sequence contains true differences from the reference genome that make it look like a suboptimal mapping. And again, we can kind of walk through a toy example. So if we have a read aligned to the Indocene reference and the taurine reference, but we come from some sort of admixed sample, some reads will map equally well to both reference genomes, no problem here. Some reads contain variation basically not found in either reference genome, so they map equally well. The real problem is if you're mapping a read that contains, say, an Indocene specific insertion to the taurine reference, it will not map well. It might not map at all. Likewise, if you have some sort of reference insertion compared to the other sequence, it again will be completely unmapped. And the question is, where does this non-reference sequence get placed? Best case scenario, actually, in this case, is it remains unplaced. The worst case is it gets mapped into the wrong location, and this will start affecting your downstream analysis. Again, this is really one of the big problems that we have with linear reference genomes, because almost every downstream genomics analysis that we do kind of implicitly assumes that we have a good alignment. You might do some filtering on the genomic alignments you have, but generally you just plug the BAM file straight into any downstream tool, get a VCF, do any popgen, whatever. And of course here, the key thing that we're trying to avoid is reference bias, which is this systematic distortion that occurs when sequencing reads are aligned to a linear reference genome, causing sequences that are sufficiently diverged from the reference to be mis or unaligned. And hopefully I'll convince you by the end that Pangenomes can mitigate this reference bias. Not necessarily that Pangenomes will eliminate this entirely, but again, Pangenomes can offer additional, more representative sequence for multiple genomes, and hopefully that will help mitigate reference bias. Likewise, when we talk about variation, it's generally going to be in something like a VCF format. If we have multiple samples that all contain slightly different copies of an insertion relative to the reference, there is no really good way to represent this in the VCF record format. So here, if you have three different, slightly different insertions, you basically have to represent this as three entirely different lines. However, jumping ahead to craft genomes, also maybe this representation doesn't look nice to everyone, but from many perspectives, this pangenome offers a more natural way that we can represent variation, avoiding some of the limitations that we get from a linear format and VCF. It's also not really a huge problem for many people, but if you talk about a linear reference genome, it's very hard to describe sequence that is before or after the linear reference genome coordinates. And many of the assemblies that we're generating today either have additional centromeric or telomeric sequence that is not present on the reference genome. And if you dig into the VCF spec, you can see that they actually talk about how to represent specifically telomeres using either position 0 or 1 past the end of the chromosome. So this is a very unsatisfying way of doing it. Where again, if you look at something like cracked genomes, pangenomes can really handle multiple starts and multiple ends to chromosomes very naturally. So you don't need to worry about how to extend past the chromosome or how to start before the chromosome. Of course, there are many more reasons that I think pangenomics helps to alleviate some of the issues that we get with linear reference genomes, but it's helpful to just kind of crack on. But here we'll go for the definition with a pangenome is a collection of genome sequences from many individuals of the same species. This is being sort of a more standard definition recently. There are a lot of problems if you look through the literature. Unfortunately, I'm part of this problem, but there are 17 articles that I could currently find that have the title super pangenome somewhere in there, which just kind of leads to this prefix inflation where if you talk about a genus, sometimes again, people use this super pangenome. But the idea is as we build bigger and bigger graphs, we have more and more samples from different species. We're going to have to start calling them mega pangenomes, hyper pangenomes is kind of useless. So really, I think the key is a pangenome is just any collection genome sequences from any set of individuals. It doesn't matter if it's the same species, same population, multiple species, genera, whatever. Certainly, there's this really great paper about Logan. So this is basically where they put every single genome that's publicly available into this sort of pangenome index. And again, if you talk about all of life into a single pangenome, I'm trying to imagine what prefix that would have. It's a bit tricky. So again, I think really a pangenome is just a collection of genomes, regardless of how they're related in terms of phylogeny. Pangenomes has also become a very common term. Certainly, if you look at conferences or paper titles coming out, they tend to include pangenome. Again, if we look at publication dates for papers, we can see there's this huge uptick around 2020. Certainly, this is when long-read sequencing became a bit more accessible. Many people had multiple genomes to work with, and pangenomes are a very natural way of working with multiple genomes. Some of these earlier papers, particularly from sort of 2010 to 2015, a lot of these papers talking about pangenomes were really talking about bacterial pangenomes, which is sort of where the term started. But this refers to a slightly different style of pangenome, basically talking about a collection of genes that are shared amongst different bacteria. So the term pangenome has kind of gone through two eras, but certainly nowadays it refers to, or more commonly refers to, whole genome sequences. When we talk about pangenome, we want to really care about what sequence is shared across the assemblies and what sequence is different across the assemblies. So you might have some definitions for something like a core genome. So this is sequence that is present across all individuals. Depending on the definitions, you have something like an accessory or a shell genome, which is sequence that's present in some assemblies, but not all assemblies. And then have something like dispensable or cloud for talking about sequence that's found in most commonly only one individual. So something like private variation to that individual. Kind of depends on the exact definition of what type of pangenome you have, which we'll cover a little bit later, but also of the quality of the pangenome that you have, which is also kind of a gray area to define. But if you consider something like having a misassembly, so one of your assemblies that you've built a pangenome from might be missing some sequence, you might take something that is actually should be present in all of your samples, so it should be part of the core genome, and it will now appear in the accessory or shell genome because of that misassembly. Likewise, if you have some sort of falsely inserted sequence, so not actually biological in origin, but technical in origin, you might start to see an inflation of sequence in the dispensable or cloud genome. So some of these topics or some of these definitions now are building larger pangenomes. They'll say something like the dispensable genome is found in 1 to 5 or several percent of the assemblies. You talk about a soft core, so it doesn't have to be present in every single assembly, but is present in almost every assembly, so 90% or more. Again, these are not really fixed in stone, and as we get to larger and larger pangenomes, we'll almost certainly have to revisit how these are more rigorously defined. Even defining variation actually in a pangenome gets slightly messy because we normally talk about variation against a reference. One thing that you commonly will talk about in pangenomes is bubbles. These are basically the regions of the pangenome that there are differences. So these are the variant loci in the graphs. There's some recent papers that came out, particularly from Hengley and some other people, where they use this much more complex definition of a variant about reference trees and paths and pairs of alleles being shared on these branch points. Again, this is something that everyone almost takes for granted in pangenomics, that how we actually define variation becomes complex because we're trying to avoid having a single reference that we always project insertions back or deletions back onto. Keeping it much more general, certainly in a graph or in the other types of pangenomes, becomes much more confusing. Now kind of coming back to the actual different flavors, so to speak, of pangenomes, there's many different ways that we can take a collection of assemblies and turn it into a pangenome. The one that I almost exclusively work with, and I think is sort of the most natural way to think about pangenomes, is what we call variation graphs. So in this type of pangenome, the nodes that we talk about are the actual sequence that's shared across assemblies, and the different edges talk about different ways to basically connect these variants. You can also talk about De Bruijn graphs. So here, instead of specific pieces of shared sequence, all the nodes in your graph are essentially k-mers, and the edges between them talk about how these k-mers are connected. Even something like a multiple sequence alignment, which again has been around for much longer, this is technically a type of pangenome. We're talking about how different genomes are related to each other in a single format. However, again, we have to be a little bit aware that the definition of pangenome is quite vague. So even something like a BLAST database is technically a collection of different genomes that we can somehow interrogate at once. So this is also some type of pangenome. And again, you can also even consider just a VCF file, something that, again, contains variation across a different set of samples as some type of pangenome. And certainly some downstream tools that we'll cover later can certainly take a VCF file in to build a pangenome. So again, it really depends on who you're talking to and what their background is for what people consider a pangenome to be. I think the key thing here as well is I like variation graphs, but there are many different approaches that require slightly different pangenomes. So it's not necessarily that one is the right type of structure for all questions that we might ask. And certainly it might even be that within one research project, you have to use slightly different versions of different pangenomes because they each have their own strengths and weaknesses. And if we jump into file formats, again, these are slightly based more on graph genomes, but we see that this transition is relatively straightforward. A linear reference genome is typically a FASTA sequence, so .fa. In the pangenome world, we talk about sort of a graph FASTA, so GFA. Indexing, we might do linearly with something like a Burrow-Wheeler transform, so BWT. Pangenomic, of course, they just take a G on, so it's GBWT. And again, for BAM and PAF alignment formats, they convincingly have GAM and GAF. It's not always this straightforward. Pangenomes, because they represent sequence in a slightly different way, might have slightly different extensions. But the key thing here is that generally any tools that you work with for a linear reference sequence will be somewhat similar to how we start working with pangenomes, even though they sound much more complex. On the bioinformatics side, they're not always that much worse. In terms of variation graphs, which again is what I have the most experience with, there's sort of three more common tools that are around at the moment. So something like Minigraph, Cactus, and PGGB. And again, they each, even though they're all variation graphs, have slightly different algorithms and slightly different use cases. Minigraph, for example, only really works on structural variation, so it doesn't build small variation into the graph. Something like PGGB, you don't even need to tell it at all which one is the reference, so it's completely reference-free. In terms of representing all the input assemblies perfectly, Minigraph, again, because it doesn't include small variation, you cannot recover the assemblies from the graph. For PGGB, you can. But on the other hand, Minigraph is very quick to run. You can run it certainly on your laptop. PGGB, certainly in the earlier days of the tool, would just run and run and run for ages, even on quite a large compute cluster. So again, it really depends on the type of question you want. If you know you're only focusing on larger variation across perhaps very diverged assemblies, something like Minigraph might be appropriate for that question. If you really need to care about SNPs and you want the most accuracy you can, perhaps something like PGGB is the best tool to use. Again, even in the few years that I've been doing this on, things have changed quite rapidly. There was the original version of Cactus is more or less a progressive Cactus. It's been almost entirely replaced by this Minigraph Cactus hybrid. And PGGB is basically a wrapper for all of the steps of building a pangenome. And there's been a ton of development recently talking about this implicit graph, so ImpG, Crush, Allwave. There's dozens of tools that come out all the time. Some of them burn bright, others just disappear. So it becomes very difficult also when you're working in pangenomics. There's very little standard, there's very little consensus on the best tool to use for any given question. You really need to play around with some different inputs, different outputs, and see what is best for your question. In terms of getting some basic statistics from the graph, so here we're probably talking about the number of nodes or the number of edges in our graph. A few different tools like Gretel or Panacus. But it's really fair to say at the moment that there's not a great way of describing the quality of a pangenome in the same way that we might do for a reference genome or any assembly. And basically, the best hope we have at the moment is if you put in good assemblies, you'll get a good pangenome out. And so hopefully in the future, there'll be some improvement in this regard. We can start to talk about the completeness of the pangenome, how representative that pangenome is of that species, that genera, whatever level we want to talk about. And again, hopefully we can assume correctness that when we build the pangenome, it will not introduce errors that were not present originally in the assemblies. When it comes to visualizing, this is a bit more of a problem. Certainly something like IGV is sort of the standard for any linear reference alignments. It's really great to be able to zoom into BAM files, to VCF, to bed files, whatever. The same isn't really possible at the moment for pangenomes, not least because how to represent this data in a pangenomic format is a bit of an open question. But there's some tools like OGI. It's really great for doing these large-scale complex graphs, and you can visualize them statically into something like a PNG. So we can see here that this is a fairly straight pangenome. There's something kind of weird happening at the edges, but in general, this looks quite linear, which is nice. On the other hand, we can view pangenomes much more interactively with something like Bandage. So this is the next generation version. And here we can really go into detail on very small regions of the graph, but it's very hard to visualize very complex graphs because the program is much slower. It's really great though. We can do many, many things with this. You can blast sequence, you can use minimap to align sequence, you can load different bed files projected into pangenomic coordinates. So we sort of have to pick, again, which direction we want to lean. If we're trying to visualize very large complex graphs, there'll be a slightly different set of tools to if we want to do a detailed investigation on a small complex region. And again, for variation graphs, we can talk about many different ways to align sequence to the graph to get something like this GAM or GAF file out. Minigraph, Graph Aligner, and most recently, VG-Giraffe has sort of started to dominate sequence to graph alignment. On the other hand, though, if we talk about the De Broglie graph style of pangenomes, there's a completely different set of tools again, something like Rope BWT3, Moni, Pufferfish. And there are many different tools, again, that align to certain types of pangenomes and will not align as well to other types. Graph Aligner is slightly better in this regard that it can align to both types of graphs, but I think certainly it's not as strong in aligning to either type of graph as a more specialized tool. And again, hopefully I haven't scared off everyone with all the detail on pangenomes. There is certainly a lot more lurking beneath the surface, but they are definitely worth exploring. And so if we consider something like a cross-species comparison, there can be a lot of complex chromosomal evolution. And so one example that I think is very interesting are these muntjacs. So Certainly, I think out of mammals, this has the smallest karyotype in one species that only has 6 chromosomes, and there's a different muntjac that has 46 chromosomes. So if we want to try and analyze these at the same time and do some sort of comparative analysis, it's quite difficult talking about a linear reference genome, because if you pick the one with 46 chromosomes and you're aligning a sample that only has 6, or the reverse, they just don't necessarily integrate that well. So the real question is, as we start to encounter larger and larger sets of assemblies, we can look across greater and greater scales of diversity. How can we handle things like different karyotypes? And one sort of way that we can get around this through pangenomes is this idea of communities. So if we do this all versus all alignment at the start of a pangenome, and then we can see where the different parts of some assemblies map to other parts of assemblies, we can build this sort of complex network. So this one was built from a handful of cattle genomes. And again, you can see that most genomes, something like chromosome 14, typically maps to other assemblies of chromosome 14. But you can see there's a couple points that are slightly drawn to a different cluster. And this might be something like a translocation or something where in one sample, chromosome 14 actually contains some sample from chromosome 6, or certainly in cattle, it's common to see between chromosome 1 and 29. And this could generalize to any number of karyotypes. It doesn't matter if your samples have the same karyotype or different karyotypes. We basically can just start by saying, well, where does the sequence map across all the assemblies? And figuring out how to analyze it then all at once, rather than having to convert to a specific karyotype as we would with a linear reference genome. And this, I think, is actually one of the most interesting papers that I read in a little while. So this is talking about recombination between heterologous human acrocentric chromosomes, where basically the idea is if you build this pangenome across all these human assemblies, most of the chromosomes separated very neatly into these clusters, but there were certain chromosomes that just exploded into this complex tangle where they would map very strongly to other chromosomes. And it turns out that this was a really dynamic part of evolution across the acrocentric chromosomes, where these centromeres shared a lot of sequence identities such that they were mapping between each other. And again, you could discover this through a linear reference genome, but this result came out very naturally from taking a pangenomic approach rather than having to sort of filter out the data in a linear approach. Another thing that looks very promising is this idea of a personalized pangenome read alignment. So again, as we talk about more and more assemblies, we talk about more and more complex pangenomes. The human pangenome version 2 that's coming out, I think soon, is something like 440 haplotypes. So this is quite a large graph. I think to download the file is probably certainly into the hundreds of gigabase. So it's not something that's that easy to work with. And a lot of that variation will not necessarily be relevant to your sample of interest. The idea here is that if you have sequencing reads that you want to align to the graph, you can basically pick up little fragments of sequence of these k-mers that look more related. And so in the case of cattle, you might have 3 different samples. You start from the same graph, and then you can remove the pieces of variation that you don't find in these fragments, so these k-mers from your sequencing reads. And now if you do the read alignment, it looks much more like a linear problem, and this becomes much more scalable. Building this personalized version of the pangenome is not quite as straightforward as it sounds. It's still computationally a very challenging problem, but this looks like a much more relevant direction that we can head into, where we can make sure we're all talking about the same original pangenome. So we are still talking about the same coordinates. We can compare is slightly different between all of our studies. And of course, we can always talk about something like QTLs, so looking for sort of interesting variation that might be related to a trait or molecular phenotype. And we can do this basically just looking through the pangenome to find regions where certain sets of assemblies or certain sets of samples look similar or dissimilar. So we did this in a paper looking for the white-headed phenotype in cattle. Again, basically just scanning across the pangenome to see where the samples that we knew had white heads looked like each other and did not look like the samples that had color heads. And we can see this nice spike on chromosome 6, which happens to be very close to a gene that has been widely proposed as being related to this phenotype. The hope here is some other people are working on this, but there's many methods from PopGen that we can start to apply to pangenomics. It's a very straightforward problem in theory, hasn't really gained enough support yet to be used widely in practice. But again, there's no reason why we can't do PopGen on a pangenome graph. It's just most downstream analysis always requires a VCF, and we just require the effort as a community to adopt some of these tools to pangenomics. But certainly it's a very natural way to talk about population genomics when you have your population in a single reference structure. The other thing that I hope will be quite useful going forward is pangenomics is quite difficult, and not all collaborators want to even deal with pangenomes. The second you start talking about graphs, they kind of stop replying to your emails. So the hope is, can we simplify this? And if we talk about just linear alignment of some sequence to any standard reference, we can use something like BWA. We just align the file, we get a SAM or BAM file, problem is over. And there's some approaches, again, like in VG-Giraffe, where we can tell it to basically align to the graph and then figure out how to convert that back into linear coordinates. So in this case, we can just tell it directly to output a SAM file, and it does all the complex stuff on the inside. I think it's fair to say that not many people really understand how a linear aligner works in detail. So it's not really that different to saying you don't know all the internal steps of how VG-Giraffe aligns to a graph and converts back to linear, but we can accept that it's been widely tested. And if we can show that there's an improvement in the alignment, I think that's a pretty compelling argument to make to any collaborators. The thing that remains to be fully quantified is if we do this pangenome round trip where we align to the graph and then convert back to linear reference coordinates, how much of that improvement survives? I think most people agree that it's more than enough to be worth it, but it's definitely not as good as working fully in the pangenome space. But convincing everyone to convert to graphs will take a little while, so this is certainly a good intermediate. Of course, there's also many challenges in pangenomics. Again, there's almost no consensus on anything. I've probably already upset some people with some of my definitions. Again, many of the tools change quite rapidly. It's very hard to stay up to date in pangenomics. If you spend all your time staying up to date, you spend no time working on it. So that's definitely not good either. The other slightly challenging thing, for example, Cactus and VG tend to be built by very similar people. So if you want to use VG-Giraffe to align to a graph, you really are best suited to use Cactus. If you want to use something like EMBL-EBI, it's built by the same people as PGTB. And so it's kind of, each tool works best with their own developers. It's very difficult to use tools across these ecosystems. Also, many public services are generally tuned to linear reference genomes. I think the final talk in the series about pangenome annotation will be very interesting. I think there are some ways that this is starting to be adopted into different services at Ensemble and NCBI. But certainly if you want to talk about how to view pangenomic sequences, it's very difficult to do this in the same way that we can do this for, say, widely accepted linear reference genomes. So another problem that I had in my pangenome comparison of builders, but if we talk about having a set of assemblies and the next day some colleague of yours generates another assembly, this is quite a difficult problem for most of pangenomics. They're generally quite expensive in terms of compute cost to construct. They're quite difficult to validate because there's no accepted tools to say this is a good graph or a bad graph. And so really, if we get a new assembly, is it ever worth just rebuilding the graph, the pangenome? And of course, many of these steps, they start with something like an all-to-all verse alignment. So if you add in a new assembly and you don't want to break the assumption that there's no reference bias, you essentially have to restart from step one. Some tools do allow you to reuse some intermediate files, but generally it's very challenging to add pangenomes or add assemblies to pangenomes as we go. Again, as we talk about how easy it is now comparatively to generate assemblies, how to routinely integrate these and keep pangenomes up to date is a very open question. And I won't go into too much detail on graph genomes because I know that's a topic for a later section, but even just defining good is quite a complex problem. If you have something like this simple graph with two bubbles, so again, on the left, we have basically an allele that is A and an allele that is G, a slightly longer allele on the right. This graph could be equally represented by this graph on the bottom. You could argue that this graph on the bottom is more simple. It's somehow more fundamentally representative. But if you want to talk about, say, changes to coding sequences, it's very hard to detect changes in, say, amino acid triplets from the lower graph because you never have three bases in a row on any of these nodes, where you do have three bases in a row on the upper graph. So this idea that there's a good graph that we should be aiming for is, again, a completely undefined topic in pangenomics. Some graphs might be better to answer some questions, and other graphs will be better to answer different questions. There's also, when you start these processes of all-to-all aligning many assemblies, you'll contain potentially many false positive hits. So typically, if you do a whole genome alignment to build a pangenome, you just end up with this completely tangled mess of all the chromosomes. If you build per chromosome, then you tend to end up with slightly tangled graphs, but not as bad. And some people are, again, exploring the idea of building little pangenomes in little windows and then stitching them all together. The problem here is that if you talk about complex events, so something like translocations or long distance, interchromosomal events, as you build these smaller and smaller scale graphs, you lose potentially all of these complex events. And if we think that these complex events are a very natural thing to study with pangenomes, again, as we try and solve the problem, we basically just end up throwing away the interesting part. So how to handle all of these false positive hits is quite difficult. And again, this is really, I think, where the plant people have it tough, because these transposable elements that you tend to have a lot in the genome basically just mean that your pangenomes are the most tangled things you can imagine. And again, this is certainly way too much detail, but you have this property of graphs in general that are called DAGs, so directed acyclic graphs, essentially meaning that you cannot loop back on yourself. So everything in the graph has to somehow proceed from left to right. Difficulty here is that the different pangenome builders, so PGGB and Minigraph Cactus, they produce graphs with very different DAGness. So Minigraph Cactus tries to enforce DAGs quite strictly, so they do not contain cycles. PGGB does not really have this constraint. So the problem is, if you consider something like tandem repeats, so sequence that is just repeated multiple times in your genomes, the way that you can build this into a graph, either having this DAG property or not having this DAG property, are quite different. And any sort of definition about allele frequency or anything like that that we can talk about in a VCF file essentially goes straight out the window when you start comparing DAG to non-DAG graphs. There's no straightforward way to define these, or even things like how far apart are these sequences. It's very complex on how to define that accurately in some of these complex graphs where you can loop back on yourself. And because we don't necessarily have a good answer on which is more biologically realistic, which graph is somehow better or more representative than other graphs, when you encounter this problem, you just have to hope that your graph is not too bad, that the alignment will fail. And again, hopefully the community will sort of stabilize a bit and say, okay, no, we really do want to enforce these types of properties. We do want to say, the pangenomes sort of have a start and end in the same way that chromosomes have a start and end. But until that really coalesces in the community, you just run into lots of graphs that just do not work with many tools because they either do or do not follow this assumption. And hopefully, as some key takeaways, pangenomes I like to define as just a collection of genome sequences. I think limiting yourself to say it's the same species or the same genus or whatever is quite limiting. We can really just talk about pangenomes now more generally as trying to analyze any collection of genome sequences at the same time. And because pangenomes can include multiple sequences from very diverse individuals, this can really help mitigate reference bias, where if we aligned to a single linear reference genome, because of things like admixture, hybridization, intergression, it might not be possible to align all of your sequence to any one single linear reference genome. This can allow us to also better represent variation rather than having to require it be projected back onto a linear reference genome. However, perhaps controversially for a webinar series on pangenomes, I think it's important to remember that pangenomes are not always going to be the answer. I think they have been applied many times in recent papers where they did not need to be. Of course, linear reference genomes are still very strong. They can answer many, many questions. I do think pangenomes can do more, but it is worth realizing that they do come with a cost. They are slightly more complex to work with, there are many problems that haven't been solved yet in pangenomics. So it's just making sure that they're worth it. Same sort of thing for T2T genomes as we talk about these sort of ultra-complete genomes. They're very topical, they look great in the title of a publication, but they're not necessarily the answer to all the problems that we had before. Otherwise, that's the end of my talk, so happy to take any questions now. Thank you.
Thank you very much, Alexander, for the wonderful talk today and giving us a comprehensive introduction to pangenomes. I'll start reading the questions, and then Alex, you can answer them. Okay, so to start with, in what ways can molecular phylogenetics be reshaped if we base evolutionary relationship on pangenomic variation rather than single reference genomes?
Yeah, so I like to avoid phylogenetics as much as possible because the concept of having a defined tree, if you follow that type of phylogenetics, is quite limiting. Again, when we talk about introgression or these complex evolutionary events, trying to force specific relationships on samples is quite complex. And genomes are quite nice because we can just say, basically for every part of the genome sequence, where is this similar to these other groups and where is it more distinct? To convert that into something more concrete, again, I think we kind of have to make some sacrifices and say, at some point, we're willing to say these two samples are more related, these samples are less related, but it may not be the case evenly across the whole genome. So I think pangenomes really help give much more fine-grained resolution to that.
Great, thank you. The next one is, why genome assembly in plants is that hard? Does it have to do something with polyploidy?
Yeah, I think there's many different reasons. I think the diploidy tends to be the higher one. Also, many plants just contain far more repeats, so particularly transposable elements, where it's very hard to uniquely identify basically which reads overlap. So you just end up with lots of possible fragments that could be your assembly, but not as clear what the correct path is.
How can bubbles in plant pangenomes help us distinguish between adaptive genetic diversity, for example, disease resistance genes, and neutral variation?
So again, there's not necessarily a straightforward way of doing this only through pangenomics. Kind of like the QTL example that I was giving, but if you have a large set of plants and some of them have perhaps the trait of interest that you're looking for and others don't, and you can identify through the graph that all the carriers of this trait take the same path through the bubble and all the non-carriers of the trait take the other path. And that's a very easy way of then prioritizing that this bubble is perhaps something a bit more worthy of your time. You can also, again, basically project the reference annotation of genes into the pangenome and say, okay, this bubble actually contains, or it's very near to a gene of interest. So we know already to start looking at this bubble and see if it matches the expectations that we would have for this impact.
Okay, thank you. Do you think that short read sequencing data can be useful for building or working with pangenomes, or is it more trouble than it's worth? Many species have few chromosome level assemblies besides the reference, but tons of short read data sets publicly available.
Yes, this is I think a very key topic, because building the pangenomes, I think you definitely want to avoid short read based assemblies. They're very fragmented, and you want the pangenome to be basically as good as possible. However, once you have the pangenome, aligning short reads to the pangenome is perfect. The main issue we have with reference bias is if you have a 150 base pair read and you're talking about 1000 base pair novel insertion, it just will not map to the reference. If your pangenome reference is more diverse, it contains these different representations, your short reads can actually align more or less perfectly to the pangenome graph. And this is really where I hope we'll be for the next several years, until long read sequencing is cheap enough to be the only answer.
Okay, thank you. Is pangenomics possible for phages? And if so, what about phages of different hosts?
Yeah, there's no reason necessarily that pangenomes wouldn't work for this. Again, part of the challenge is a lot of the people who build pangenome tools tend to work in the human community. So if your genome is not about 3 gigabases in size and doesn't match sort of human levels of diversity, you tend to have to play around with the parameters a lot more than you would probably like. So certainly for phages, I think you would have to customize the parameters significantly. The main challenge also of building pangenomes is if there's not enough sequence similarity between the inputs, do you really want to try and force it into one structure? That's the other side of the question.
Thank you. How do we standardize for genetic data saved in different formats? Example, chromosome positions versus SNP IDs. Should we change all to VCF? Thanks for the awesome presentation today.
Yeah, so this is kind of another buried skeleton. So when we talk about the reference genomes, I think a large part of the community's resistance to changing reference genomes is it is a lot of work changing from one coordinate system to another. Certainly converting from linear to pangenome is possible. If we have a fairly stable pangenome, we can talk about, node 700, position 40. That's where the SNP happens. And we can be sure that we're talking about the same position, the same sequence in my study and your study. Again, once we update the pangenome, the coordinates will be different. We'd have to talk about, updating all those coordinates. So this is definitely a big problem, particularly I think when we talk about annotation, how to do this in an agreed way on a pangenome. It is possible, but it's definitely another open problem.
Okay, thanks. Should we dereplicate or not when we analyze pangenomes? Some species strains were sequenced again and again, which causes the bias for data.
Yeah, I wish I had the answer for this one. We, at the moment, at least in my work, we don't have this problem too much. We have a large collection of assemblies, but generally on slightly different animals. The problem is if you build this pangenome graph and 50 of your assemblies are the same and 2 are different, you will suddenly make the same variation look far more important than it is. Many of the alignment techniques kind of look towards very frequent nodes. So again, if you have many inputs that are actually from the same strain or the same clone, you might make that look more important than it should be. And again, that's kind of a choice of how the aligners work, but that will be a bias that kind of filters through all the analyses that you do.
Okay, great. What would be the best approach for gene set comparison using the annotations from each assembly separately or re-annotating the pangenome to ensure consistency?
I am probably leaning towards using each annotation and trying to merge them together. Kind of like the example I had for the graph where you can split one large bubble up into smaller bubbles. I really don't know how pangenome annotation will work. I think there's many different ideas, but you can't annotate a single base. So this idea that you could annotate the graph as a structure, I don't think will ever be possible. Annotating all the assemblies in the graph and somehow merging when the sequence is shared, I think that makes a lot of sense. But again, that's just a very fancy linear reference genome intermediate.
So the next question, they want to confirm if accessory or dispensable genomes contain new genes or variants of a gene.
Really depends on your definition. When we talk about, at least in the context of variation graphs, if you talk about something perhaps in the accessory or the dispensable part of the genome, again, we could be talking about just a single base. If you want to talk about the gene level, you'd have to adapt these definitions slightly. Certainly, if you contain a single mutation of a gene, that might completely change its function. It might have no effect. So how to sort of functionally categorize that into, is it a variant of the same gene or completely novel? Again, you have to just be very careful on your definitions and make sure that whoever you're talking to shares those definitions.
How can we tell whether bubbles in a pangenome represent real functional diversity or just artifacts purely through computational methods?
Yeah, again, at the moment, we effectively can't. Um, the hope is that nowadays we can test if our assemblies are real, that they contain biological diversity and not technical diversity or technical artifacts through various K-mer quality variants. We can talk about sort of realigning the short reads or long reads to make sure the coverage is even. If the input assemblies are all good, we kind of assume that the pangenome is good. Certainly there can be these cases though for repetitive elements that if you basically find a false positive because these sequences look quite similar across different chromosomes, you might end up with something that looks like variation, but really is just, unrelated homology.
How did the emergence of pangenome affect, if it did, the definition of species?
So I'm not sure if it's really affected the definition of species in that sense. I think we certainly now don't need to be quite as strict on the definition. Certainly, building up to the genus level for these pangenomes tends to go quite straightforwardly. Even if there are karyotype differences or quite different genome sizes, it can be handled quite naturally. Really, I think the key at the moment is, do your sequencing reads align well to whatever input reference you want to do, if it's a linear reference or a pangenome. Because certainly you can align something from the same species that's actually very distant to the reference, at which point you run into a ton of problems. Or you might have two very similar species that you can align to the other reference with no problem. So I don't want to redefine species, but really it's the average nucleotide divergence to the reference. That's what you should be worried about when doing alignment.
How do you think pangenomes may contribute to understanding non-coding gene regulation?
I don't know if they'll particularly have much effect, especially about that. Certainly, I think when we talk about pangenomes now, we can look at much more complex types of variations, so particularly larger structural variants that, again, in the VCF format tend to get or more complex to represent. I think in a graph, they can be represented sort of the nested variation in much more detail. So if non-coding and these other complex regulatory regions sort of are governed by more complex processes, I think this might help with that. But ultimately, we just can understand variation and represent that variation better, not specifically anything to do with coding versus non-coding.
Right. Does the human pangenome contain mitochondrial DNA genes?
I believe so. I'm not 100% sure, but again, this is kind of the tricky part because most human chromosomes are quite large, so the parameters that they've tuned tend to be for large chromosomes. At least in cattle, the mitochondria is only 16 kb, so comparatively tiny, and in fact gets explicitly removed by most standard pipelines. So again, you run into this issue when you're trying to deal with very small chromosomes and very large chromosomes. Maybe in that graph, they basically just use the reference plus some small variation, but I'm not sure entirely.
Okay, thank you. Next one is an interesting one. How many genomes do we normally require to make a reference pangenome?
Yeah, I think the answer is anywhere between 2 and infinity. Really, the key is you just want to be able to represent the diversity that you might find relative to whatever research question you want. So if you're mapping a very small population that has a very low effective population size, you obviously will not need that many genomes. If you say want to try and map, any possible human variant without any bias, you would then need a very large pangenome to make sure that we can represent, the many different, populations that you might find across the world. So it really depends on essentially what you're using it for, of how much of that diversity do you really need to capture.
Okay. Thank you. We just have 1 minute left, so probably we can take this one last question. For pangenomic QTLs, is it possible to perform permutations to define the statistical threshold, or would it require such a large amount of computational power that it is currently not feasible?
I think there are more efficient techniques that are coming out that would make this possible. Um, Certainly, one of the problems that you have in these very complex graphs is if you just look at the structure of the graph, you might have many different bubbles side by side are not that are not all equally likely to make like a real haplotype. So if you just do a permutation on the graph, you can end up with very unrealistic, not biological in origin haplotypes. So how to handle that correctly, you then sort of jump back into having multiple linear references. It gets a bit confusing, but I think there is hope that we'll be able to do essentially GWAS on a graph directly. We don't need to worry about converting back and forth, but we're not there yet.